Alison, for inviting me to talk today. It's always nice to come back to Brunel and see lots of friendly faces. But I'm now at the University of Brighton. I'm Emma Ross, and my interest is in voluntary control of muscles. So essentially, how the motor cortex, the part of the brain that's responsible for volitional movement, how the motor cortex drives the muscle, and in particular, how exercise affects that neural drive to the muscle. And in particular, further, how exercise-induced fatigue interrupts that neural drive or affects it. And my PhD under the supervision of Alison and another doctor at the physio school was to look at the neural control of respiratory muscles, the voluntary control of respiratory muscles. But since then, my interest has gone into more skeletal muscle in the limbs, so locomotor muscles responsible for movement during exercise. And that's really where I'm going to come from with regards to my talk, looking at how signals that arise in the locomotor muscles during exercise feed back information to the central nervous system that then play a role in the mechanisms of central fatigue. So I'm going to kind of run through some of the basics and unravel that title in a bit more detail during my talk. So I didn't want to gloat, but I will. I just wanted to point out that this is where I'm from this morning. This is my office, just down the road from Eastbourne Pier. And this was my run route yesterday, and beautiful at this time of year. And for those of you who knew me 4 years ago when I left Brunel, this is also what I've been up to since I left. So this is my little boy, Bodie. Plenty of distraction from work, as you can imagine. So when Alison asked me to come and talk about why does inspiratory muscle training work, how are the respiratory muscles involved in afferent feedback, I gladly accepted the opportunity to talk. And I sat down to write the presentation, and I had lots of questions because, as I said, my interest really is about how the locomotor muscles signal to the brain during exercise about fatigue and cause fatigue. And so I had lots of questions about how we might use the evidence that we have about afferent feedback from locomotor muscles to the central nervous system, how we might use that to hypothesize about the role afferents from the respiratory muscles might work in the process of central fatigue. So I had lots of questions going around in my head about how we might make that relationship. And so I thought I would base this presentation around a number of questions which you might come up with given the title of my talk. So the first one might be, Emma, what is central fatigue? And I'll just touch on what we define as central fatigue to start the talk off. And I'll also mention quickly how we can measure central fatigue to kind of put into perspective some of the results I'm going to show you later in the presentation. The main part of the talk will be focusing on a number of studies which try to unravel how afferent feedback, how signalling from the locomotor muscles is involved in central fatigue. Unashamedly, I'm going to be borrowing a lot of the work that's been done by previous Jerry Dempsey students who have worked with Dr. Roma before, so Marcus Amann and his colleagues. My most recent work has been looking at how things other than afferent feedback might affect central fatigue. And those are in situations where you might have a lower inspired oxygen fraction in hypoxia, or you might have arterial hypoxemia, or in very high heat conditions, where actually the brain itself has far more control over output independent of feedback from the periphery. So my research has kind of moved over into that area. So I'm going to borrow some of the really nice research that's come out of Marcus's lab about how afferent feedback is involved in central fatigue. And because we're ultimately talking about inspiratory muscle training, I'm also going to try and relate how training can induce changes that will improve our central fatigue response to a given bout of exercise, and touch on then how that might relate to inspiratory muscle training. All the way through, I'm going to try and bring in some of the respiratory muscle stuff, although some of it will just be hypothesizing from what we know about locomotor muscle to another skeletal muscle, the diaphragm, and the accessory muscles of breathing. And then if I have time at the end, I should really check the time. If I have time at the end, I'll just briefly touch on the work that we're currently doing in our lab with inspiratory muscle training in collaboration with Alison. So a good place to start this talk, and Lee has already touched on it, so he's given me quite a nice introduction. What is central fatigue? And we have to start with what is fatigue. And fatigue in this sense, because fatigue is a complex phenomenon and means different things to different people, clinical definition of fatigue will be different from an exercise-induced fatigue definition. So in the context of excuse me, my work and this presentation, we're talking about a reduction in the muscle's ability to generate force, full stop. That's how we classify fatigue. And we can measure that by getting someone to do a maximal force-generating maneuver, whether it be an elbow flexion, leg extension, and measure the muscle's ability to produce force during a maximal effort. And if that has been decreased, we define that as fatigue. And exercise-induced fatigue is characteristic by the fact that that impairment is reversible by rest. So again, in clinical presentations of fatigue, they might be more chronic. So when we have volitional movement, we obviously have areas of the brain which program movement, coordinate movement, and plan movement, and they input neural information into the motor cortex, which is ultimately the part of the brain which drives the muscle. That signal then is propagated through the corticospinal system, and that usually monosynaptically, but sometimes via a network of interneurons, synapses onto the motor neuron, which synapses at the neuromuscular junction to the muscle. So this is the pathway of neural propagation during volitional movement, such as exercise. And fatigue is an impairment that causes output here to be decreased. But it can happen because of a change at any point along this pathway. So when we use the term peripheral fatigue, what we mean is there's been an impairment in the muscle's ability to produce force because of changes that have happened at or distal to the neuromuscular junction. So the excitability of the muscle membrane is changed, the contractility, the ability of the muscle to shorten, or something at the neuromuscular junction to do with neurotransmitters has been altered. So that's what we might term peripheral fatigue. Central fatigue is a progressive decline. And we've already mentioned that fatigue is a progressive phenomenon, not a sudden onset. It's a progressive decline in output or drive from the motor cortex to the muscle. So it's impairments that are happening upstream of the neuromuscular junction. And upstream is quite a lot of events. So it's obviously to do with spinal propagation of information, corticospinal, intracortical, what's happening in the motor cortex itself, and then supracortical, what's happening in all other parts of the brain. So it's obviously, like I said, fatigue is a complex phenomenon, and just looking at neuromuscular fatigue is particularly complicated. Some of the ways in which we unravel at least where the impairment is occurring is by using muscle or nerve stimulation, and most of the studies that I'm going to show you today will utilize nerve or muscle stimulation to try and interrogate the neuromuscular system. So we can stimulate the muscle itself to look at just the muscle's ability to turn an electrical signal into a mechanical event. We can stimulate the motor nerve to look at neuromuscular junction transmission and then contractility. We can stimulate somewhere along the level of the spinal column to look at spinal and perhaps reflexive control of movement. And we can stimulate at the motor cortex of the brain, all non-invasively, to look at output from the motor cortex. And so if we can measure things at different sites along this pathway, we can start to build a picture about where the impairments might be happening, which lead to an ultimate reduction in the muscle's ability to produce force. So how do we measure fatigue, and how do we measure central fatigue in particular? This is quite a classic setup for us in our lab and for many others who are looking at the same phenomenon. And it's comprised of a number of things. So in a lot of the studies I'm going to talk about, we're interested in knee extensor function, so the quadriceps. So in this case, we're looking at quadriceps function. We have someone tethered to a load cell to perform an isometric knee extension. So when we want to test how much force they produce, we ask them to do a maximal knee extension. They can't go anywhere because they're tethered to the load cell, so all the force they're producing is measured as torque or force about the ankle. We can measure the activity of the muscles by placing EMG electrodes on, and Lee's alluded to that already. You can't see, because it would be rude to, that we have a small electrode placed in the femoral triangle, which via very brief electrical pulses will stimulate the femoral nerve, which elicits a muscular contraction in the quadriceps. And that's our peripheral stimulation technique. Then we also have here transcranial magnetic stimulation, which is stimulating underlying neurons in the motor cortex. That signal is propagated to the muscle, and we record the mechanical or electrical event at the level of the muscle. And this tells us a bit about output from the cortex. And some of the data we might generate from these tests, firstly, this is a force trace, and this is a muscle twitch. So if you can imagine someone's at rest, they're not producing any force, and then we deliver a little zap to the femoral nerve, and the muscle contracts, and we get a rate of force development, and a rate of relaxation, and an amplitude of the twitch, and then the muscle goes back to rest. This is completely non-volitional, so we're overriding the person's motivation and volition to perform effort. And this is a really good measure of peripheral fatigue or contractility of the muscle. How well is the muscle contracting in response to an electrical signal, regardless of anything upstream? So some of the studies that I'm going to show you today are reliant on interpreting data that arise from this twitch force, is what we call it. If we look at responses from the transcranial magnetic stimulation, we record the electrical event. This is an EMG trace recorded at the level of the muscle through EMG electrodes. This big artifact here is the stimulation. That's where I discharge this magnetic coil, which the rapidly changing magnetic field excites any underlying tissue. In this case, very excitable tissue is underneath because it's overlying the brain. It doesn't cause any discomfort or pain because unlike electricity and electrical stimulation, magnetic stimulation bypasses any pain receptors on the surface of the skin. So you don't feel any pain, but you do excite and set about an action potential, if you like, in the underlying neurons. And you can see the time it takes for that signal to reach the muscle of interest by this latency, and the amplitude of this evoked response can tell us about the excitability of this pathway, this brain-to-muscle pathway. These data are just showing how we determine output from the motor cortex or voluntary activation. So one of the techniques that we've validated more recently in our laboratory, along with work I was doing with Lee, was to look at using transcranial magnetic stimulation to measure voluntary activation of the muscle. That is, how fully your brain is able to drive your muscle. So your muscle obviously has a capacity to generate force, but that's limited by how much your brain can drive that muscle to produce force. And most people who are healthy and fairly active can produce around 90% of their full potential to generate force in their muscles. So voluntary activation is around 90%. And after exercise, as we become fatigued and a central fatigue process is set in, voluntary activation decreases, and you might only be able to activate 75% of the potential of your muscle. So we validated a technique where we could measure voluntary activation using transcranial stimulation. And what that allowed us to do was to determine whether output from the motor cortex was optimal for that maximal effort or that contraction. And when we validated it, we determined that it was sensitive in a fresh muscle to record levels of around 90% activation in fit, healthy subjects, and after a fatigue, drops to around 75%. And that's one of the markers we might characterize central fatigue. So if we're talking about peripheral fatigue, we might talk about changes in quadriceps twitch force, if we're looking at the neck sensors. And if we're talking about central fatigue, we might talk about changes in excitability of the pathway. That's how well the electrical signal has been propagated through that pathway. And we might talk about voluntary activation or cortical output, how well the motor cortex was driving the muscle. And it's now really well established that central fatigue happens. It's really well established that central or exercise, and particularly more continuous, prolonged, high-intensity endurance type exercise, elicits changes in the excitability of the motor cortex. and in output from the motor cortex, and in the brain's ability to drive the muscle fully. And over the last couple of decades, a lot more attention has started to be paid to the role of central fatigue. Because if we think back to the origins of fatigue research, it was very much fatigue is what happens in the muscle. When there's metabolic disturbance in the muscle can't contract as well and you produce less force. And no one really thought about what's happening upstream of that until a little bit later. And then our ability to drive that muscle became important. So lots of well-documented evidence that central fatigue in healthy people performing vigorous exercise happens. But it is very task dependent. And so when I say it happens, and we've already talked about why doesn't fatigue happen to everybody in terms of the respiratory muscles. Well, central fatigue doesn't happen all the time. If you're doing very brief anaerobic power type efforts, you won't see any decrement in output from the motor cortex. Even if you do repeated intermittent sprinting activity like you might find in games play, we are currently finding it quite hard to elicit central fatigue even over 40 minutes of very kind of vigorous, repetitive, high-intensity intermittent work because there's lots of rest. But in things like prolonged running, we consistently see really nice changes in output from the cortex. And we've done things like measure central fatigue after treadmill marathons, and we see nice classic kind of responses that immediately after exercise, depression in the output from the brain, inhibition in output from the brain, and that recovers within sort of 4 hours. Sometimes some people take a bit longer to recover. So that's central fatigue taken care of as an introduction, and really why I wanted to give you that background was because we're going to talk about afferent feedback and its role in central fatigue. So this is a 2010 points-counterpoints debate in JFIS between Marcus and Mann and actually a number of other people joined in about whether afferent feedback was important in limiting exercise performance and whether it played a role in central fatigue and in fatigue in general. And one of the reasons that this generated quite a lot of interest was not only because it touches on the work of Tim Noakes, who always, if there's an opportunity to have a say, he'll have a say. Always provokes lots of really thought-stimulating ideas, but lots of people contributed to this debate. Is afferent feedback important? And today I'm going to show you some evidence that suggests that I certainly think, and that the evidence is strong, that afferent feedback is important in limiting exercise performance in healthy individuals in a normal environment. So not really hot, not really cold, not hypoxic, just your normal kind of high-intensity endurance exercise environment. Afferent feedback is important. But I think one of the reasons why it stimulates so much discussion is that it's really hard to test. For a long time, the brain and the output from the brain was really hard to test. You had to do it electrically, and stimulating the brain electrically is no fun for anyone. Certainly I wouldn't let anyone come near my brain with some electrodes. But so we had, then with the advent of magnetic stimulation, we were able to start to learn about output from the brain. But certainly neural traffic, and neural traffic that's coming from everywhere, all the muscles involved in locomotion and movement, and going to goodness knows where in the central nervous system, is hard to pin down. And so a lot of the evidence we have is based on clever research design, but not necessarily direct evidence, because I don't think we really, in the intact exercising human, have the ability to do that yet. In the animal models, probably it is the case we could start to get to the bottom of it. So what is afferent feedback? Well, both Alison and Lee have touched on afferent feedback. Afferent feedback is neural information that's sent from the periphery back to the central nervous system, in essence to inform it what's happening at the periphery. And we have afferent information about everything that's happening, as you know, about temperature, about blood flow, about oxygen and carbon dioxide, and lots of things. And afferents fall into different groups that are characterized by their discharge properties. So you have some afferents, groups 1 and 2, which have a relatively low tonic discharge rate all the time, and then they have a spike when something happens. for example, the muscle is stretched vigorously or a lot of tension is suddenly produced in the muscle. And those that afferent spikes in its feedback to the central nervous system, and then probably the signal is dampened off again. We have other afferents, groups 3 and 4, which are sensitive to lots of noxious stimuli at the cellular level, and when we talk about exercise, specifically about the metabolic status of the muscle, but also to do with pain arising at the periphery. And those afferents tend to ramp their discharge in accordance with, for example, during exercise, the accumulation of metabolic byproducts. So rather than having spikes, they ramp their discharge and their feedback up as these stimuli become more important. And what we do kind of understand is that these feedbacks will have input to the central nervous system at a variety of levels. So as we know, some of this feedback is reflexive. So we don't necessarily have any opportunity to intervene with the stretch reflex. The tendon tap is just in voluntary spinal reflex. And some of this afferent feedback is going up to the motor cortex, and it might affect the excitability, or it might affect neurotransmitter control in the cortex. And some of the afferent feedback is bound to be going to other areas of the brain. That's purposely vague because I don't think we really know too much about where, but certainly areas upstream of M1, the areas that feed neural information into the motor cortex. And as I said, we don't really have a map for the exercising humans specifically about where that afferent feedback goes, but intuitively we would expect that some of it's going to the motor areas and some of it's being delivered to the premotor areas, and some of the information might be being integrated at a sensory level or in areas that are to do with concentration and attention, because we know those things are also affected by fatigue. So the afferent feedback could be propagated to any of these areas, but what we're interested in is what effect that has on output from this part of the brain. So wherever it's going and however it's being integrated, is it having an effect on the corticospinal projections which come out of the motor cortex down to the muscle? So one of the first studies which asked the question about, does the metabolic state of the muscle inform the central nervous system about itself, and does anything happen as a consequence? And Simon Gandiva over in Australia, using TMS for a very long time, very elegant studies, looked at this in quite an isolated experiment. So they just used one muscle group, and they used the biceps. So they had people doing a 2-minute maximal voluntary contraction, which was their model of fatigue. That's a good model of eliciting lots of peripheral fatigue, because what you do in a 2-minute MVC is you occlude quite a lot of the blood flow. So you don't allow too much delivery, and you don't allow too much removal of all of the stuff that's happening during exercise. So a 2-minute MVC elicits quite a severe degree of peripheral fatigue. So they had people doing a 2-minute isometric, so they just anchored their hand down and got them to pull up for 2 minutes. Try it, it's really horrible to do that, 2-minute MVC. And what they measured was, in the white data points, the force. So if we're seeing fatigue, we're obviously going to see a drop in force. They measured via TMS, stimulation of the brain, voluntary activation. That was output from the cortex, and that's the black data set. We'll just concentrate on the bottom graph mainly, but just to let you know that these data were about the excitability within the cortex itself, and that will become more important in a minute. So they obviously did some baseline measurements. They saw that force, this is percent of initial, so that's 100, and voluntary activation was around 95%. Good voluntary activation from your fit, healthy, rested subjects. And then they started the 2-minute MVC, and as you can imagine, during this 2-minute MVC, force decreased, even though effort was still maximum. So even though here force was very low compared to initial force, the person still believed they were performing a maximal effort. At the end of the 2-minute MVC, the researchers inflated a cuff around the arm to hold the muscle ischemic. So what they were trying to do was hold it in a state of metabolic stress that was occurring at the end of exercise, but without the stress on the motor system to drive the muscle. So they wanted to just hold this muscle here at the end of the 2-minute MVC for about 5 minutes in metabolic derangement, if you like, and see what happened. Because if feedback was important, that metabolic stress being prolonged would obviously feed back information to the central nervous system, and it would affect the measures they were making. If it wasn't important, because the motor system was resting, because they weren't contracting, then clearly there would be some recovery. So, held the muscle ischemic, held it in a state of metabolic stress after the 2-minute MVC, and their measures of corticospinal excitability, which had increased during contraction, because that's what happens. Your motor system becomes quite excited during contraction because you are driving it. So the increase that they'd seen during contraction stopped as they went to rest, but the levels of corticospinal excitability were the same when the cuff was inflated in this metabolically stressed muscle as before in the fresh muscle. Sorry, down here. So corticospinal excitability, the excitability of the brain to muscle pathway, recovered after about 2 minutes of exercise, which is what we would expect if we didn't have the cuff inflated. Measures of force production and voluntary activation were still decreased when this muscle was held in a state of metabolic stress. So what we started to have was a disassociation between corticospinal excitability, one of our measures of brain to muscle function, and cortical output, voluntary activation. So what this first shows us is that something to do with the muscle being in a metabolic state of derangement was causing these measures to remain depressed, because when the cuff was released and the muscle could recover metabolically, blood flow was restored, there was recovery of those measures. So something to do with the muscle being in a metabolic stress was signaling for these measures to be decreased. But it wasn't anything to do with excitability within the cortex, so that was allowed to recover. So this suggests that any changes which are happening as a consequence of afferent feedback are happening upstream of the motor cortex, because excitability within the cortex itself seemed to recover fine, even when the muscle was still metabolically stressed. But what didn't recover was our ability to drive the muscle fully. And that can be affected by neural information that's sent to the motor cortex that then subsequently is driven to the muscle. So we're talking about afferent feedback to areas upstream of M1 that then affect the drive out of M1. So this was the first evidence, really, that holding a muscle in a state of metabolic stress did actually signal to the central nervous system to do something, because it affected output from the central nervous system. But it was a while before we had any other kind of advances in answering this question about afferent feedback. And about 10 years later, in around 2006, Marcus Amann started publishing his work on afferent feedback. And in particular they were hypothesizing that peripheral fatigue was a tightly regulated variable, and that the CNS, the central nervous system, regulated peripheral fatigue. And they had done some initial studies which had showed them that peripheral fatigue never exceeded a certain level. They couldn't fatigue people any more than this particular level. And so they said that maybe this is a critical threshold, and maybe something's regulating our ability to push ourselves beyond this critical threshold. So their first few papers set the scene for that, and then they did some further work which tried to tease out some of this information. So this study from 2008 had a control condition where performers were doing a 5K time trial. So a nice ecologically valid test. Go as hard as you can, do 5K as fast as you can. And beforehand and after, they made measures of force generating capacity and also muscle contractility via stimulation of the femoral nerve. And that was their measure of peripheral fatigue. In another condition, they wanted to pre-fatigue the muscle. Because they thought that we could only push our muscles to a level of fatigue that was set, they hypothesized that if we pre-fatigue the muscle and then we make you do a time trial, surely you're going to accrue more fatigue. You can't just reach a set level of fatigue and then do more exercise and not fatigue anymore. So what they wanted to do was severely fatigue the muscle before exercise to see if they could push people past this critical threshold of peripheral fatigue. So they had people do an exercise test to volitional exhaustion at around 80% of peak power output. So that's a hard test, and they found there was substantial quadriceps fatigue when they got off this bike. And they had about 5 minutes rest, and then they had to do the 5K time trial. You'll see from subsequent studies, these people like hurting people in their labs. And so they did this severe pre-fatigue trial, and then they went on to do the time trial. To give you some kind of dose-response perspective, they also did a moderate pre-fatigue trial, where they pre-fatigued the quadriceps a small amount before they then went on to do this time trial. And then they made measures of peripheral fatigue and force-generating capacity before and after each of the cycles. So what did they find? Well, let me draw your attention first to mean power output during the 5K time trial. There is no surprise that when you come off this bike knackered, you don't do as well in this trial. Everyone here will not be surprised by that. So power output produced during this performance trial was less if you severely pre-fatigued the quadriceps. Fine. But what was really interesting was that the level, I've highlighted in purple here, of fatigue was the same at the end of the 5K time trial, regardless of how much you had fatigued the muscle in advance. If we look at data about how much we fatigued the muscle in advance, in the severe fatigue trial, we had about a 35% decrement in quadriceps function. So they got off this bike with about 35% contractility decrease. They then did 5K time trial, and they still had 35% contractility decrease. So this supports the hypothesis there might be some critical level of peripheral fatigue that we attain, because however much you start with, you can only go to a certain level. And even when they started with a small amount of peripheral fatigue, they then accumulated enough to get them to reach this critical threshold. Marcus and Mann made measurements of EMG during the cycle trial. He used that as a surrogate measure of central neural drive. There are some issues associated with extrapolating that information, but let's call it our global measure of neural drive, as he did. And you can see that neural drive, neural output from the CNS was reduced, which subsequently resulted in less power because of this pre-fatigue. So now we're getting more support that peripheral fatigue is a strictly regulated variable. If it's strictly regulated, who's regulating it? It's not a who, it's a what. What's regulating it? It must be the central nervous system, because the central nervous system is driving it to contract. If the central nervous system is driving the muscle to contract and it needs to regulate it, it has to know about the status of fatigue in the muscle to know when to reduce neural drive to limit peripheral fatigue. So feedback has to form some part of this puzzle. The central nervous system has to know about the fatigue state of the muscle to be able to regulate it. So there was some evidence here that afferent feedback was playing an important role in reducing central neural drive during exercise to protect, if you like, and Marcus has certainly used that word before, protect the muscle from catastrophic damage, from damage that will perhaps be irreversible or unrepairable. And their conclusions were that the CNS processes neural feedback from the exercising muscles and regulates exercise by adjusting output from the brain. So then they went on and were a bit more invasive in their research design, because they actually decided to block afferent feedback. Rather than just assuming afferent feedback was happening, they wanted to intervene with it. So they actually had 3 trials, but the main difference between their conditions was that during a 5K time trial, afferent feedback was either intact, normal, just get on the bike, ride 5K as hard as you can, or it was blocked by an opioid analgesic called fentanyl. in the development of this research design, this lab had to go through a number of chemical analgesics to see which ones would block afferent feedback without affecting efferent flow. So what I mean is, can you stop signaling back to the central nervous system without affecting how the nervous system drives the muscle? Because some of the analgesics they tried, the people got on the bike and they couldn't produce as much power. It was affecting afferent and efferent signaling. So they came on to using fentanyl, which they provided some evidence blocked afferent feedback but not efferent drive. And they also had a trial which was a placebo. So it was a fairly well-controlled trial, saline infusion. This was fentanyl into a spinal infusion of fentanyl. They had a saline infusion and just a control condition, no tampering with the subject whatsoever. And they had them ride 5Ks as hard as they could in a random fashion. And these are the data they generated. So on the bottom graph, you can see power outputs. The white triangles are the experimental condition, the fentanyl, the afferent block. The black triangles and the circles are placebo and control, i.e. no tampering with the afferent feedback. What you can see here, especially if you're a cyclist, is an aggressive pacing strategy. These people set out on a 5K time trial, and their power outputs within the first K was significantly higher than when they were adopting a maximal but achievable pacing strategy. So this is a really aggressive pacing strategy. They suffered because they weren't able to maintain that aggressive pacing strategy, and actually overall the time taken to perform 5K wasn't different. And you can see this is again their surrogate of neural drive. Yes, it's very high in the beginning of the 5K time trial to produce this extra power, and then it drops off. One of the things I like about these data is it reminds us that afferent feedback isn't switched on and it doesn't switch down neural drive. Afferent feedback is probably a process that develops from the very beginning stages of exercise. And in particular, if I just, swoop over this for a minute and look at the rate of metabolic turnover, if you like. This is capillary blood lactate, so a surrogate measure of kind of metabolic processes during exercise, was higher in this hugely greater peak power output at the beginning of exercise. And so what this was suggesting was that afferent feedback might not necessarily be important, or it might not be solely important telling the central nervous system about the magnitude of metabolic stress, but it might tell the central nervous system about the rate at which we are metabolizing and causing metabolic stress. So not just the magnitude, the rate as well, because if we haven't got afferent feedback telling the central nervous system that we are turning over energy quite quickly, it's happily just driving away as hard as you can until actually the muscle becomes so fatigued you can't produce as much force. So this afferent feedback is important from the beginning stages of exercise as well as when the magnitude of metabolic stress becomes great. When they looked at the fatigue measurements, this is the control trial, this is the placebo trial for all of the subjects and the mean with the white data points. They saw comparable levels of peripheral fatigue at the end of the 5K time trial, anywhere between 25 and 40% decrement in contractility. But at the end of the fentanyl trial, they had severely greater peripheral fatigue. This supports the hypothesis that if peripheral fatigue is a tightly regulated variable, and you take away the information that you're feeding back to the central nervous system, it can't regulate it, and it lets it just keep going, and you experience more peripheral fatigue. So again, these data were suggesting that afferent feedback is important for informing the central nervous system about the rate and the magnitude of metabolic and other noxious events at the cellular level to prevent fatigue from accruing to beyond what these researchers call critical threshold. By blocking afferent feedback, a centrally mediated brake on drive from the brain is released, and it allows excessive levels of peripheral fatigue to be tolerated. So one of the most recent papers from this group actually goes further and looks at what they call the ensemble of afferent feedback. So how does the nature of the information, the afferent feedback, and where it's coming from affect the CNS regulation of peripheral fatigue. So they had a single limb exercise study, and they had a whole body exercise study, and they matched the workload relative to the peak power achieved in each exercise modality. And what they hypothesized was that if afferent feedback is coming from a very specific location, let's say a single exercising muscle group, such as during single leg knee extension, the afferent feedback wouldn't be as great to the central nervous system, and the central nervous system will be able to cope, whether that's in a sensory term or in neural drive term, cope with that feedback better than if feedback was very diffuse from its locations. Whole body exercise will generate feedback not only from the main locomotor muscles, synergistic locomotor muscles, also, as we know, many other events to do with neuromuscular and cardiorespiratory going on during whole body exercise. What they found was that exercise time was 36% longer in this single leg exercise, even though that, relatively speaking, they were meant to be exercising at the same intensity. And that quadriceps twitch force, i.e. the level of fatigue, was allowed, if we're using their terminology, to be greater in the single leg knee extensor exercise than in the whole body exercise. That is, that afferent feedback from a diffuse set of signals perhaps caused more regulation by the central nervous system than when feedback was coming from a small localized muscle mass. Quadriceps rather large, but a localized muscle mass. And this group started to use the word sensory tolerance, because something I haven't mentioned about afferent feedback is its effect on sensations and perceptions of effort. And I'm hoping Caroline will much more eloquently than I can describe some of those relationships. But certainly, afferent feedback isn't just about altering neural drive. That will have consequences for how hard you think you're working. And also, afferent feedback into sensory areas of the brain will have implications for how hard this feels. So they've started to use terminology that's slightly different. The sensory tolerance limit is influenced by the magnitude of the ensemble feedback. So I've been talking about locomotor muscles, and we are trying to get to respiratory muscles. So how can the respiratory muscles be implicated in this picture? Well, first of all, Lee introduced the concept of the metaboreflex, that work in the respiratory muscles causes sympathetic discharge, that causes vasoconstriction in the limb muscle, less blood flow to the limb muscle, more fatigue, because more metabolic stress. If you've got more metabolic stress, you've got more afferent feedback, and you've got more effect on central nervous system function. So the metaboreflex itself just augments peripheral fatigue, and if we are to look at the evidence provided by the Aman group, that would suggest that we would then elicit an augmented central fatigue response. But also, I quite like this idea of ensemble feedback. It's logical to me because we have obviously lots of signals being sent back to the central nervous system during whole body exercise from the locomotor muscles, but also about arterial oxygen saturation, about the rate of metabolism, but also about fuel levels, about mobilization of fuel, about body temperature. And these are all integrated, and we kind of perceive them as a sensation. And why can't the respiratory muscles be part of that ensemble feedback? And if you start working them harder, they're skeletal muscles. We know that quite often they fatigue, so they will probably contribute to that ensemble of feedback. And so if we imagine all of these to be making a noise as part of this orchestral ensemble, then the muscles that drive flow in and out of the lungs will also contribute to that ensemble feedback. And obviously, the brain is our conductor, receiving all the information about the sound and then giving information out about how that should then continue. So I quite like that ensemble of afferent feedback theory. Still lots of work to be done to show how that will hold up. And so I haven't really got time to go into lots of detail about how this has implications for training, but that's okay because it's quite a simple story. If you train, you become a more metabolically and cardiorespiratory efficient exerciser. You can exercise at higher intensities for the same degree of effort, or if you exercise at the same intensity, you can go for longer. If you become more metabolically efficient, you're generating less metabolic stress at a muscle level, and therefore you're having less afferent feedback and less control of the central nervous system to decrease drive to the muscle. So training, in its traditional sense, will just attenuate this afferent feedback response or delay it. And we've done some work in our labs looking at high-intensity training models, in classical endurance training models, and how that affects central fatigue. And as a general rule, all this data is showing you is that two different exercise training modalities, high-intensity training or classic endurance training, generate similar responses, i.e. We are less fatigable after training. We have about a 50% improvement in our fatigue response. And we have about a 20% or 15 to 20% improvement in our peripheral fatigue levels after a given bout of exercise when we've done in this case, 3-week training regime. So we're less fatigable, we've got less peripheral fatigue, therefore we should have less afferent feedback, therefore we should have less central fatigue. So the training story is kind of just a natural extension of that. So in conclusion, because I know I'm running a bit close to time, the research that's coming out of the afferent feedback literature during high-intensity exercise in a normal, healthy individual, in a normal environment, no hypoxia, no extreme heat, that's a totally different story to do with the central nervous system. Locomotor fatigue is a tightly regulated variable, it seems. The central nervous system, therefore, might regulate neural drive to the muscle to prevent it exceeding a critical level of fatigue. And afferent feedback is suggested to be the mechanism via which this information is relayed to regulate this variable. And some of the relationships with the respiratory muscles are that the metaboreflex will augment this peripheral fatigue, afferent feedback, central fatigue phenomenon. And the respiratory muscles might contribute to an ensemble of afferent feedback that's firing information back to the CNS, which then allows it to more tightly regulate its drive to the muscles that are causing work. So we started off with some questions. We answered them, hopefully. Central fatigue is how the brain regulates its output to muscles. How do we measure it? Well, Marcus and Mann uses some clever research design, the pre-fatigue studies, the afferent blocks, and we can use brain and muscle stimulation to look quantitatively at excitability and drive in the central nervous system. How is afferent feedback involved? I hope I've shown you some strong evidence that feedback from the muscle can be integrated at a central nervous system level to reduce central motor output. How can training affect that? Well, if you reduce peripheral fatigue via training, you can reduce afferent feedback and central fatigue. Training also improves your voluntary control of muscle and co-activation of synergists as well. And can you tell us about your current IMT work, Emma? No, I probably don't. Oh, no, sorry. The last one was, what was that one? How can all this be applied to muscles of breathing? We can dampen the respiratory metaboreflex, and we can reduce its input to this ensemble of noise during exercise. And can you tell us about your current IMT research? No, I don't have time. So all I'll say is we're using IMT in kids who have neurodegenerative disease to do with cerebellar ataxia. They have poor coordination of muscle, and we hope to improve that coordination of breathing muscle to improve cough effectiveness in a clinical population. And so I'd be happy to talk to you about that either in the question time or afterwards, just mingling. Sorry for going on a bit longer. I'll leave you with some interesting facts about the brain, and thank you for your time.